Hello kids, this is Tolbert's biology class, genetics. This is lesson 4. Lesson 4. Tolbert's biology, lesson 4. Cell division. Write this in your word maps. There are two different ways that cells divide, mitosis and meiosis. You need to know that mitosis is what we call regular cell division. This is how you grow larger, bigger, make repairs, things like that. Whereas meiosis is how you make gametes. Gametes, as you already know, are the egg and the sperm cell. And in that particular type of cell division, you cut the chromosome number in half. Cell division. In meiosis is the form of cell division which results in reducing the total number of chromosomes from the diploid number to the haploid number, so you cut it in half. This only happens in the sex cells, that is, the sperm and the ovum. Mitosis is the form of cell division which results in the formation of identical daughter cells. So like when your skin repairs the skin, each cell will make a cell that looks exactly identical to the first cell. Okay? This is where we have tissue growth and repair occurs throughout the body, except, of course, through the sex cells. Genotype or gamete, part 2. Genotype is the actual genes, so the letters. Okay, geno is the actual genes or the letters, so capital letter or lowercase letter. Contains 2 copies of a gene, the same, big or small. Here, you've got big A, little a, 2 capital B's, big C, little c, 2 capital Z's, little j, capital J. Gametes contain only 1 copy of a gene, so there are no repeats, big or small. Looking at this, all the way down here where you have A, C, Z, B, E, F, there are no copies, so there are no big letter and small letter. They're just individual. So the best way is for us to determine what the gametes would be for this possible genotype. Capital A, lowercase a, and big B, big B. Here's how you do it. You do it through this process called FOIL. First, outer, inner, last. First, outer, inner, last. In this example, we have capital A, lowercase a, capital Y, lowercase y. The first two are A, capital Y. The outside would then be outside. The inner would be there and there, and followed by the last is being there and there. First, outer, inner, last. Okay, so you can put this in your word map first. So using the genotype of big A, big B, little b, you've got the first, outer, inner, last. So the real, actually, only possibilities are big A, big B, little a, big B. So here, we want to tell the difference if it's a gamete or a genotype. So, remember, a gamete has no repeats, where a genotype is going to have repeats. So, the first one is the genotype. Gamete. Genotype. Gamete. Genotype and gamete. Listing all the possible genotypes for this particular combination. Using FOIL, you should be able to do this. Those are the possible: first, outer, inner, last. Okay? You've got the first, outer, inner, last. Genes. Chromosomes contains units of inheritance called genes. Usually they occur in pairs, one from each parent. Some traits are inherited directly from one set of genes. Some traits are inherited through several sets of genes. It was presumed that there was over 100,000 sets of genes, but this is not true. For extra credit, if you find a report about how many actual genes that we have, you print out a copy, bring it to me, and I'll give you extra credit. Dominant genes. The genes are expressed whether they are dominant or recessive. It has the power to overshadow the recessive ones if they're dominant. Some examples are white-faced cattle, droopy ears in pigs or swine, polled cattle. Polled means they have horns, or the black coat in the white Angus cattle. The gene that is overshadowed by the dominant gene is called recessive. Recessive genes can only be expressed themselves with the absence of the dominant gene. So the only time you see them is when they are two lowercase letters together. Here are some examples. Albinoism, dwarfism, those type of things. If both letters are the same, use the prefix homo. Zygous. Homo means the same. If both letters are different, you use the term heterozygous. Hetero is the prefix that means different. The term genotype means the actual genetic makeup. The actual genetic makeup is the letters used to represent that trait. The term phenotype is the physical characteristic, or what it looks like, as it being tall or short, the actual appearance. Punnett squares. So in this example that you have in your notes, this is some of the stuff that you'll need to use in order to build this Punnett square. We're going to figure out how many calves are black and how many calves are red. The dominant gene is the capital letter, lowercase letter is the recessive. So let's come up with how many of the genotypes will be dominant, how many of those guys will be recessive, the ratios. Here's how you build the Punnett square. You put one on the top, the other on the bottom. You could put dad on top. I guess here's how I've done it. One of the parental units on top. One of the parental units on top. Big B, lowercase b, the other parental units on the side, and you just simply match it up. Booyah. Booyah. Booyah and booyah. And you continue to do this. This B can go right over here. Boom. 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 And there, here. That's how you build it. So we're going to practice with capital R, lowercase r, big T, so you can decide where you want these pieces to be placed. I'm going to put one on one side, one on the other. So I'm going to put capital R, lowercase r, and two capital T's here. And let's go ahead and put them together. T versus R, and T, sorry. I said T and I wrote one other thing. And T versus lowercase r. Here I've got a capital T and a capital R and a capital T and a lowercase r. Let's look at the ratios. How many of them are dominant, are homozygous, or both of these are dominant, and they are heterozygous, which means they're two different ones. So 50% of them are heterozygous dominant. All right. And last but not least, the results. So this tells us how many of each, and this tells us the ratio. Thank you for listening.